I'm Jeff Kometsa, Chief Business Officer, and I'll take you through a brief introduction of our company. Our management team has over 150 years of experience in the pharmaceutical industry. We are led by our Chairman and CEO, Dr. Da-Jun Yang, who, with Dr. Xiaomeng Wang, another co-founder, have dedicated over 20 years studying apoptosis. We have built a world-class pharmaceutical company. Our innovative platform is focused on protein-protein interactions, which play a critical role in cellular function. We focused on 4 key proteins that regulate 3 different apoptotic pathways. Ascentage has over 400 employees worldwide dedicated to one mission, making this a oncology-focused, patient-friendly biotech company. We have worldwide IP, and for the rest of this presentation, we'll focus on 5 key compounds, which are in 15 trials and 10 INDs across the world. This slide provides a view of our core assets in our portfolio. Our HQP-1351, our third generation tyrosine kinase inhibitor, has completed its pivotal study, and we will submit our NDA to the Chinese government later this year. The rest of the molecules have made tremendous progress over the last year, all of which are progressing into phase 2 clinical trials. So let's take a closer look at HQP-1351. This is our third generation tyrosine kinase inhibitor aimed at the BCR-ABL mutation in CML. Our pivotal studies are complete, and we expect the data readout mid-year this year. We will file our NDA to the CDE in June of 2020. The data is predicated on a phase 1 study that demonstrated very high activity in specifically T355I mutations, but all patients in both chronic and accelerated phase CML. What distinguishes 1351 from other tyrosine kinase inhibitors is its phenomenal safety profile. We've had no cardiovascular, cerebrovascular, or other typical AEs that are associated with other third generation tyrosine kinase inhibitors like ponatinib. We also have a profile that enables patients to stay on therapy for a long period of time. We have patients on therapy now for over 3 years. Only 2 of the 101 patients in our initial phase 1 discontinued therapy, and that was due to progression of disease. Recently, we've had several key milestones. We received orphan drug designation and fast track approval for our ongoing study conducted at MD Anderson, which expects to be our registration program moving forward. On slide 8, we show you the summary of the grade 3-4 AEs, along with the responses by specific criteria for the chronic phase CML patients. Of note, the major cytogenetic response, which was the primary endpoint of the trial, achieved an 82% response rate in the T355I mutations. If you look at the overall responses in the patients, both wild-type patients as well as the mutated patients all did well on HQP-1351. So moving on to APG-2575. This is our select BCL2 inhibitor. It will be the first follow-on to venetoclax. We have enrolled over 7 cohorts in our phase 1 dose escalation trial. What makes us different is we escalate on a daily basis versus a weekly basis. And to date, we have seen only laboratory TLS, no clinical TLS, which has put a change in the dosing regimen for venetoclax. So far, we had no DLTs, and the MTD has not been reached. Of note, of the 21 patients I've treated, 6 of the 8 CLL patients have all reached objective response criteria. If you check the milestones below, recently we've had several 1B2 trials approved by the FDA, one for relapsed refractory CLL and the other for relapsed refractory Waldenstrom's. Additionally, we have filed an ODD for Waldenstrom's and are in the process of submitting separate protocols for multiple myeloma, TPLL, and non-Hodgkin's lymphoma. Currently, there are 2 phase 1 dose escalation trials underway in the US and in China. In the US trial, we've enrolled 21 patients, 8 of whom have CLL, and of the 8, 6 have reached objective response criteria. The patient on the right is a patient that presented with a white count of 120,000 and bulky adenopathy. By the end of cycle 1, the patient's peripheral count has normalized, and their lymphadenopathy by cycle 5 was reduced by 90%. Besides these trials, we've recently received approval for a relapsed refractory AML trial in China, as well as 2 trials in the US that are phase 1b/2s that will be used in combination with standards of care. The CLL trial has already been initiated with our first patient enrolled a month ago. So all BCL2 selective inhibitors are not created equal. Here we try to distinguish the difference between 2575 and venetoclax. Besides the dose escalation or the ramp, ours is weekly versus monthly for venetoclax. We also have a difference in a PK profile, which enables us to hit the target at similar picomolar concentrations, but get out of the body. It's a hit and run target. Once you saturate BCL2 and cause apoptosis, you don't need to hit it any harder. So by getting out of the body faster, we potentially lower the amount of neutropenia. Additionally, we have 2 metabolic pathways, potentially avoiding drug-drug interactions. And this is specifically useful when you're thinking about AML, where you have a lot of drugs like ketoconazole, posaconazole, that are typically used for prophylaxis. And we do not have the same drug-drug interactions. So let's take a look at APG 1387. This SMAC mimetic is a dimer blocking both IAP and XIAP. We've conducted 3 dose escalation trials and demonstrated the drug to be very safe. We've also seen some interesting signals pertaining to modulation of the tumor microenvironment. So we quickly opened up a 1b/2 trial where we've enrolled only patients who have been resistant or refractory to checkpoint inhibitors. And we've combined 1387 and Keytruda, and we're seeing an objective response rate of about 13%, which is early, but 50% of the patients have disease control rate. And of the evaluable patients, 13 patients are still ongoing, showing continued improvement in their responses. Additionally, we've finished a phase 1 trial in hepatitis B in Chinese patients. And we've demonstrated that by targeting the host cell and generating apoptosis, we can significantly reduce both DNA levels as well as surface antigen levels. The results of this trial have been so encouraging that we've quickly opened up a phase 2 trial in China in combination with nucleoside analogs. And this will be a sequential trial where we give a month worth of 1387 followed by several months of nucleoside analog. So in slide 12, we have an example of one of the 4 PRs that we've seen in the trial. This is a non-small cell lung cancer patient who was PD-1 negative and has a history with other primary cancers and being sensitive and intolerant to chemotherapy. They were enrolled in the trial and within 3 cycles had a 50% reduction in the tumor. And after 5 cycles has a 70% reduction. This patient continues on therapy. And it's just one of the 4 examples that we've had demonstrating the activity of this combination. On slide 13, we show you some of the highlights from our hepatitis B trial. Patients received 4 weekly doses of 1387, followed by a 6-month evaluation. And the segment on the right, you can see the dose cohort responses in DNA levels. Sufficient to say that not only did we see both DNA levels, but surface antigen level reductions. We're encouraged by these results and have initiated a phase 2 trial combined with nucleoside analogs. So turning to APG 115, which is our MDM2 p53 inhibitor, we've conducted 2 dose escalation trials and have identified the phase 2 dose. We've also demonstrated single agent activity as well as a strong signal of tumor microenvironment modulation by MDM2 inhibition. We initiated a phase 1b/2 trial. The results of the phase 1b are there. And in 19 patients, we've seen 1 CR, 2 PRs, and 6 ongoing stable diseases for a disease control rate of over 64%. In the phase 2 trial portion, we've already enrolled 26 patients in 6 different dose cohorts. Additionally, we have initiated trials in AML and have filed for orphan drug designation. So slide 15 demonstrates the CR patient who was enrolled in the 1B trial. This is an ovarian cancer patient, typically a cold tumor and typically non-responsive to checkpoint inhibitors. This patient also had an ATM mutation. They were heavily pretreated and failed multiple lines of chemotherapy and other experimental trials. The patient was enrolled on study and within 1 month started showing tumor regression. And by cycle 3, they were in a PR, and their responses continue to improve over the next 4 cycles. By cycle 5, they had reduced their CA 125 levels to almost normal levels, and by cycle 6, they were at normal levels. This patient remains in CR. So let's look at APG 1252. This is our dual inhibitor of both BCL2 and BCL-XL. BCL-XL is a unique target. Not only is it highly expressed in various forms of lymphoma, but it's also highly expressed in patients who are chemorefractory. So in diseases where chemotherapy is a standard of care, non-small cell lung cancers, and say gastric cancers, patients that have failed first-line chemotherapy have high levels of XL expression. In the US, we finished a small cell lung cancer trial and demonstrated single-agent activity in small cell lung cancer, as well as other advanced cancers. What makes us different from the other dual inhibitor that's in the clinic is that we've avoided, by design, irreversible platelet toxicity. I'll go into more of that in the second slide. Additionally, we have ongoing trials in non-small cell lung cancer combined with Tagrisso in patients who have become resistant to Tagrisso and have an EGFR mutation. We also have a trial ongoing in small cell lung cancer combined with paclitaxel. Additionally, at MD Anderson, we're running a trial in myelofibrosis in patients who have progressed on ruxolitinib. We filed for orphan designation and expect to have that early mid-year. Well, in slide 17, we make 2 points here. First, we demonstrate that there is no irreversible platelet toxicity, and this is by design. We use a pro-drug approach so that the drug is only metabolized in the tumor tissue. On the right-hand side, you can see the results of a small cell lung cancer patient that received a PR and stayed in PR for over 20 cycles. The patient died of pneumonia unrelated to their primary cancer. Ascentage is at the forefront of apoptotic targeted therapies. We are the only company worldwide that has drugged 3 of the major apoptotic pathways with 4 different assets. All of our assets are available for partnering globally, and we look forward to discussing these opportunities with you. Please give us a call,